Welcome to this next session of our Neuroblastoma Parent Symposium. This session is Neuroblastoma Drug Development Strategy, New Therapeutic Targets and Strategies. And it's being delivered today by Dr. Lucas Moreno, Clinical Director of Pediatric Oncology and Hematology at Vall d'Hebron Hospital in Barcelona, Dr. Jan Molenaar, Principal Investigator from Prinses Maxima Centre in Utrecht in the Netherlands, and Dr. Stephen Du Bois, Director of Experimental Therapeutics at Dana-Farber Boston Children's Cancer and Blood Disorders Centre. And with that, I will hand over to Dr. Lucas Moreno to get the session going. If you've got any questions, please use the Q&A box at the bottom, and we'll have a few minutes at the end to get through as many as possible. Over to you, Dr. Moreno.
Thanks, Nick. Thanks very much for the very kind invitation. And I am very, very honored to be in this symposium. So as you mentioned, we will be sharing the presentation between the three of us, and I'll starting with the neuroblastoma drug development strategy. I'll start with the strategy that we've got. So I'll briefly show my disclosures, and I'll try to briefly cover today what is that we call NDDS, New Drug Development Strategy for Neuroblastoma. What was the need for it? We have up till now done 2 meetings, and it's a dynamic strategy that continues. And what are the outputs and the future strategy that we have? So it all started nearly 10 years ago with the need for a concerted strategy. Those of us working in neuroblastoma back in 2010, 2011, we were all aware that patients with high-risk neuroblastoma had poor outcome. There were no targeted drugs clinically available. It was just the year that the results of the anti-GD2 study by the Children's Oncology Group and Dr. Yu were reported, and these weren't even introduced into frontline still or still not widely available, and none of the targeted drugs had reached the clinic. Many drugs were being tested in the laboratories in neuroblastoma models, but none were making it into clinical trials. And indeed, in general, for pediatric cancers, there were very, very few phase 1 trials across the globe. The companies were starting some pediatric trials following the regulation, mandates and incentives by the regulation, but most were not focused on neuroblastoma biology. So it all started with, as many things that you will have heard in this conference, it all started with an idea from Andy Pearson that was working at Royal Marsden in London then, and he got together a group of us from Europe, from 3 particular groups, from ITCC, the consortium that develops early phase trials, from CIAPEN, the consortium that works on neuroblastoma, and from ENCA, which is a research project to improve the outcomes of childhood cancer. And we all started this strategy, and we went to organize the first meeting, which was very positive and novel. It happened in London in 2012, and it was one of our first multi-stakeholder forums. We had clinicians, we had scientists researching in neuroblastoma, and we had regulators. And we focused on 6 main areas that where work was necessary to move forward in the field: on preclinical data evaluation, on targets and drugs that needed to be prioritized, on the population that should participate in early clinical trials of neuroblastoma patients, on that we wanted to push for a mechanism of action driven drug development process, considerations on early phase clinical trial design, and randomized phase 2 trials. What happened? We worked on the 6 areas, and we agreed that targets should be evaluated on a systematic way. So for every target, we should always look at details of the target, of the actual target and its presence in neuroblastomas, activity in vitro, in cells that are growing in the lab, in vivo, in murine models, and some other information about biomarkers, about how can the drugs become, how can the tumors become resistant, and how to combine the drugs. We used this systematic way of evaluating targets to evaluate 21 targets into high, intermediate, or low priority for neuroblastoma. We focused on relapsed and refractory neuroblastoma as a population for trials. There was a group of people that focused on this mechanism of action-driven drug development, and together with people in both in Europe and in North America. Since then, some regulatory changes have happened in both sides of the Atlantic, with more focus for targeted drugs being developed in pediatrics, regardless of the disease, the specific disease happening in adults. And the key example is crizotinib, that was a drug, an ALK inhibitor developed for lung cancer, that was not developed for children by the company because lung cancer did not occur in children, even though it was really important for neuroblastoma and other pediatric tumors. Regarding early phase trial design, we pushed for novel designs to be incorporated so that pediatric trials could be faster. And we also wanted that there were more randomized phase 2 trials. There were none at that time in relapsed neuroblastoma. And for example, since then, there are 4 studies have been started or completed in relapsed neuroblastoma with randomizations included. We went on to the second meeting, which was even better. It happened 5 years ago in Cambridge, 5 years later in Cambridge. And we not only had clinicians, scientists, and regulators, but we had 3 major additions, which really made sense and made a lot better the initiative. We had representatives from pharma. We had North American colleagues. We had many of them, but led by Steve Du Bois and Julie Park. And we also had parents in the meeting. We focused more on targets. The rest of the areas that I mentioned were better suited. So we did focus on epigenetic targets, for example, TERT or ATRX that were relevant for neuroblastoma, on targeting MYCN, and we also reviewed other targets with the idea of prioritizing drugs that were of 2 areas of work. So one is drugs that were ready for clinical evaluation, and the other was targets that were interesting but had no available drugs. And this is a very crowded slide on the output, but this is just to say that on the left, we started with evaluating 40 different targets in many areas, including those epigenetic targets. And we came up with 3 drugs, 3 groups. One, priority targets that had agents already in pediatric trials. So for those that were already in pediatrics, we wanted that the early phase trials were faster to complete and faster to transition to phase 2 and 3 if they were successful. We also had another category that was priority targets with agents that were in adults in the clinic, but still not in pediatrics. So for those, the actions was to push the companies so that they went into pediatric trials, to persuade the companies to facilitate pediatric trials. And then there was another group of priority targets with no agents in clinical development, which were some targets very relevant for pediatric neuroblastoma with very good preclinical data, but that unfortunately there were no good drugs to target these targets. So the efforts should focus on drug discovery efforts, so on the universities and companies to try to develop new medicines for these targets, which were a long way from being a reality in the clinic. This is another way of looking at the output in terms of the clinical development of the drugs that made the way from phase 1 to phase 2 to phase 3. And again, this is a crowded slide. The ones that have an arrow are the ones that continue in development. The ones that don't have an arrow, their development was stopped or is on hold for the moment for pediatrics. But fortunately, all of these are pediatric trials of interest for neuroblastoma, and drugs are moving forward into phase 2, some into phase 3, and we'll hear some examples. But it's I think it's positive. It's a positive overall output that we are seeing drugs being moving forward. So in conclusion, the strategy is great to bring everyone together, clinicians, scientists, pharma, regulators, and parents from across the globe to build on a common strategy, which is based on identifying those targets and drugs of interest very early in the development, sharing the priorities with all the communities so that pharma, regulators, everyone knows where the efforts should go. We still need to speed up clinical development, the drugs move faster, and facilitate a concerted effort to bring more drugs into the clinic. And for some other drugs to promote drug discovery efforts. We are already working on NDDS3, which unfortunately will have to be virtual, but we'll continue to work on it. And then as many new drugs are reaching clinical development and moving forward, still, I think it's a both positive and negative balance. We are seeing very good advances, but still some drugs for very good targets, like that we know in neuroblastoma and that we find in the molecular profiling programs, still they're far from having a clinical trial or being available. So with this, I'll finish and hand over to Jan Molenaar to continue on with our presentation.
Thank you, Lucas. I will share my screen. So, and open the presentation. Yes. Okay, so indeed, thank you, Lucas, for this first part. My name is Jan Molenaar. I'm from the Prinses Maxima Center in Utrecht, the new pediatric cancer center, which is open now for 2 years, where we've united all pediatric cancer treatment and also the research in one center. It's really working out very fine. And I would like to continue on the path of Lucas, and where he explained that we would like to proceed with real targeted therapies, but to make that work, we need to know what is wrong in the tumors of the patients. So therefore, we need precision medicine programs. And I would like to talk you through what that stands for. First of all, you know, of course, that neuroblastoma are heavily treated with this high-dose chemo, surgery, chemo, stem cells, and then the GD2 and retinoic acid. This is actually the protocol in the Netherlands. There's several protocols used worldwide, but in short, it's extensive, aspecific therapy that is given to these children. And this, in at least a portion of patients, high-risk patients, this is effective at first. But then in a lot of patients, the tumor finally starts growing again, or after a period, it relapses. And that's when the real problem starts, because these relapses are very hard to treat. And as you know, the treatment of the primary tumor is actually quite a specific. It's chemo, it's toxic substances, radiation, and that is really hitting the tumor, but not good enough, because the tumor is coming back and it's damaging the child severely with all the acute side effects, but also the long-term side effects, as you know. And what we'd like to find then for the relapses, therapies that are specifically targeting the tumor and leaving the child unharmed. And we would like to identify these therapeutic options. These will first be used in relapsed tumors to see whether indeed they work. And then, of course, in the end, we would like to put them up front. So for these type of therapies, we have a few basic principles in precision medicine, where first we have to understand that cancer is caused by DNA aberrations. So something is damaged in a cell, in the DNA, causing the cell to grow faster. Then there's additional events occurring. And then through clonal outgrowth, you get these hugely malignant cells that keep on growing and spread throughout the body and damage everything. And then eventually can kill the host. And if we want to find therapies that are specifically working in these tumor cells, we should identify therapies that are specifically targeting these DNA aberrations and thereby killing the tumor cells and leaving the child unharmed. And this is actually a picture of taken right at the right moment when the US was testing precision bombing. And so this is what we were striving for. So for that, we have the precision medicine programs, which have certain steps to identify these DNA aberrations that we can target. So first, we take a biopsy and we isolate DNA and RNA. You can ask the question, well, why do we need to take a biopsy? Because most of the patients have already had a biopsy at the primary tumor. Can't we do the analysis in that biopsy? Well, Well, for that, we have to understand that if we look at the tumor prior to treatment, so in this stage, before the treatment starts, this looks like this. And this is a figure, a circus plot, which indicates all the genomic aberrations that are present in a tumor. You don't have to understand, but it's about the principle. You have the mutated genes, deletions, structural variants, and this is prior to treatment. And this is the same tumor, but at relapse. And as you can see, there's a lot of additional events in that tumor. So if we want to identify genomic aberrations that we can target, we should look at the tumor at relapse. And that's why we have to take a new biopsy. So we take a biopsy, and then the RNA DNA is isolated, and then we do the sequencing and data analysis. And sequencing, most of the precision medicine programs currently do some kind of sequencing analysis that covers the large part of the genome and the large part of all the events that can occur. And that varies a bit between the institute, but it's more or less the same. You try to cover the complete genome. We analyze this using the R2 bioinformatic platform, which has been presented yesterday, as I heard. When we have the analysis, then we continue to our tumor board, and the tumor board we discuss with the experts, the pathologists, the clinicians, the phase 1/2 trial experts, and the biologists, what type of aberrations there are, and we try to prioritize these aberrations. And this prioritization works in a way that we identify genomic aberrations. For instance, here an ALK mutation, F1174L, with a corresponding, it's an actionable event because there's a compound that works in that against that genomic aberration, which is lorlatinib. So it's a druggable event, which is a true genetic change. It's a direct drug target. There's a lot of evidence, specifically in neuroblastoma, so we have a very high level target. When we, for instance, have MYCN amplification, there's no compounds specifically targeting MYCN. So it is a genomic aberration, but it's not a direct drug target. It's more a prediction biomarker, which indicates that tumors might be more sensitive, for instance, aurora kinase inhibitors. So then we end up with an intermediate score. And when we have, for instance, ATRX with ALT, then we know the genomic aberration, but it's actually no compound that specifically works good in these tumors with ATRX deletions. So we have a genomic aberration, but it's actually non-actionable. So in that way, we classify the events, and then finally we give a treatment advice to the treating physician, and we advise whether there's trials available or whether there's compounds that you can give off-label or on basis of compassionate use, and then we advise how these patients can be treated and which trials these patients can go to. I think it's really important to understand that this is the aim is to get patients with relapsed tumors in the right trial, and eventually we would like to use these compounds in an upfront setting. The chances of fuel creation after this treatment is still very, very, very small. So I think that is important to realize. So there's several programs running currently in Europe. These are the programs shown here. So the MAPIEX is in France, and the INFORM is a large project in Germany, which also profiles for several other countries. We're running ITER. There's a big group in Australia running Zero. Danish and the English are running programs. In the US, it's coupled on the precision medicine trial programs that are run there. So this sounds all very great, but there's of course a few bugs, and one of the most important ones is that in about 50% of the patients we can identify good actionable events. But there's also a group of patients in which we can't identify actionable events. For instance, there's ATRX, but we don't have a compound against ATRX, and there's other genomic aberrations, 72 gain, but we don't have specific compounds against it. So for that group, there's several initiatives going on to identify additional events, immune profiling as well, but also there's a program which we are doing and several other labs are running currently to not only take the biopsy for DNA isolation, but also generate an in vitro model, so grow the cells, and then actually use those cells to test a high amount of compounds and see which compounds do work. So independent of the genomic aberration, see just which when we throw over about 200 compounds that are currently available, and then see what works. So in that way, then we try to optimize and also be able to give an advice for patients who do not have genomic aberrations that are actionable. So this whole project gives an advice and identifies actionable events. And Stephen Du Bois will continue to the next step. Then how do we go from there to the trials, and how does that work? I will stop here. Stop sharing, and then Stephen will take over.
Thanks so much, Jan. I appreciate the opportunity to talk with this group about how we try to move agents that are identified through the NDDS into the clinic. And as Jan pointed out, there's this phrase we use, actionable events. You can have an actionable event if there's a mutation or another genomic alteration in the tumor that matches with a specific drug. But if you don't have a genetic change, there's not going to be an action. But likewise, if there's not a drug, there's not going to be an action, even if there's a genetic finding. So a big part of my job is to try to move novel agents from the laboratory into the clinic and conduct what is called a first-in-child clinical trial, meaning a phase 1 clinical trial where it's the first time we're using a novel compound in children with cancer. And recently we became very interested to look at the landscape of these first-in-child clinical trials to understand what is the lag time between the first time a new agent gets into an adult human and the first time a new agent gets into a child. We all know in this field that there are real challenges in getting novel agents into the pediatric clinic. And so we had a very industrious medical student working with us who actually cataloged the time from the first in human clinical trial to the first in child clinical trial. And each row or each bar on this plot represents that time or that lag time to get into the pediatric clinic for 126 FDA-approved oncology drugs. And what we showed is that there's really a lag of about 6.5 years from the first in human study to the first in child clinical trial, which I think we all would agree is too long, understanding the importance of finding novel agents for neuroblastoma. So I thought I might share a couple of vignettes with the group that talk about or really give you a sense of how we move an agent that's been identified by the NDDS into the pediatric clinic, and how that sort of shapes up against this median of 6.5 years. So the first class of drugs I'll talk about are the BET inhibitors. This is a group of drugs that that work by impacting how genes are turned on and off in neuroblastoma cells and in normal cells. And so a collaborator here at Dana-Farber named Kim Stegmeyer did a screening study where she looked at a wide range of adult and pediatric cancer cells. So each of these circles represents a different type of cancer. It's a cell line growing in a petri dish in the laboratory. And she looked to see if we use a BET inhibitor on all of these cell lines, which ones are more sensitive, so those over here to the left, and which ones are less sensitive. And what she showed are that these red cell lines seem to be particularly sensitive to BET inhibition. And these red circles all represent MYCN amplified neuroblastoma. She then looked at a mouse model of MYCN amplified neuroblastoma and showed this is a survival curve where 100% of mice are surviving up until this point and then start to die of their neuroblastoma. And what we see here is that the mice treated with the BET inhibitor had a much longer survival compared to the mice treated with salt water, basically. And so these two findings made us extremely interested in the potential for BET inhibitors to have an impact in children with MYCN amplified neuroblastoma. Oops, that went too fast. These inhibitors were also thought to be important in a wide range of other cancers. And as is standard, there were a large number of clinical trials in adults. And you can see a wide number of drug companies developing these inhibitors with a range of different names here in all of these different diseases. Many of us in the field, when we saw the data from Dr. Stegmeyer, tried very hard. I probably met with 50% of the companies on this list trying to get one of these compounds into the pediatric clinic. And for a variety of reasons, we were not able to succeed for quite a long time. So in fact, there were no pediatric trials despite having data back as far as 2013. And so ultimately, we were very lucky to see a grant opportunity from an organization called Stand Up to Cancer, a philanthropic organization that makes grants to researchers. And they had partnered with a drug company, Bristol Myers Squibb, to provide access and funding to do clinical trials of Bristol Myers Squibb compounds. And so it was only through this type of an innovative collaboration between a funding partner and a drug company that we were finally, about 6 years, so at about the median time for all of the other first-in-child clinical trials, we were finally able to open a first-in-child clinical trial of this drug, BMS 986158. And so the clinical trial includes an initial cohort where we're looking at unselected patients with neuroblastoma, but also with other diseases, because in parallel with the research from Dr. Stegmeyer, we've also seen that this class of drugs may be important for other pediatric tumors. But I'm a neuroblastoma researcher, and so I wanted to make sure that we also carved out specific slots for patients with MYCN amplified neuroblastoma. So we have what's called an enrichment cohort here that allows us to enroll patients with MYCN amplified neuroblastoma or MYCN amplified other pediatric tumors that have MYCN amplification. So this study opened last year. It's currently open at 4 centers. We're the lead center here in Boston. It's open at the Hospital for Sick Children in Toronto, University of Michigan in Ann Arbor, Duke University, and it is planned to open at Cincinnati Children's and at Seattle Children's. So this is a clinical trial that we've been able to develop with support from both Stand Up to Cancer and from Bristol Myers Squibb. The other vignette I wanted to share is something you've heard a little bit about already from Dr. Goldsmith in her comments about NANT clinical trials. You heard about why we have been interested in aurora kinase inhibitors and our prior success with a less selective inhibitor called alisertib that showed nice activity when combined with chemotherapy in a prior NANT clinical trial. However, that clinical trial also showed that the alisertib together with chemotherapy was rather toxic, with a really fairly extensive effects on the bone marrow. And so we weren't quite sure what the next steps were for aurora kinase inhibition until we learned about this new compound, LY3295668, which I'll just call 668. And so this is a compound being developed by the drug company Eli Lilly. And just like Dr. Stegmeyer's work, Eli Lilly conducted a screening study to try to understand which types of cancers might be most impacted by this Aurora A kinase inhibitor. And so instead of looking at the types of cancers, they looked at the genetic features of those cancer cell lines to understand what genetic features were associated with sensitivity to this compound. And one of the top predictors of sensitivity was MYCN amplification. That, of course, got us very excited. They separately did a screen evaluating more than 560 cancer cell lines, and neuroblastoma was among the most sensitive of the histologies that were evaluated. They went on to do some animal studies and showed regression when this compound was used in neuroblastoma models. And they had fairly recently completed the phase 1 first in human clinical trial and showed that the drug had a favorable safety profile. So with that information, we were able to work with Lilly to help them develop a trial that they are running. It's an industry-sponsored study, but with input from the NANT consortium that you've heard about in North America and the ITCC consortium that you heard about in Europe. And so this first-in-child clinical trial is open for children 2 to 21 years of age, exclusively with neuroblastoma. So we're really following the preclinical data that all point to neuroblastoma as an important histology with this compound. Patients need to have adequate organ function, like adequate liver and kidney function, adequate blood counts. They can't have received prior Aurora kinase inhibition. And at least for now, the drug is only available as intact capsules. So children need to be able to swallow intact pills, which is obviously an issue for some of our younger patients with relapsed neuroblastoma. The trial will look initially at the drug given on its own, but given what we saw in our prior NANT trial of alisertib, a different Aurora kinase inhibitor together with chemotherapy, we're very keen to see how this agent performs when given together with chemotherapy. And so how does this trial stack up against our median of 6.5 years from our prior analysis? So the first in human clinical trial of 668 began in May of 2017, and the first in child clinical trial, I'm happy to say, opened in June of 2020. So that's about a 3-year lag, and on this plot would put us right around here. So I think we're doing a little bit better, at least in terms of this example of getting an NDDS agent into the pediatric clinic. And I thought I would just share with you some of my thoughts about what it really takes to get an early first-in-child study off the ground, rather than having promising targets sit and wait and have a longer lag time. So I think it obviously takes a committed industry partner who is interested in following the preclinical data that all point to neuroblastoma as a disease of interest. It takes academic investigators like myself to advocate and provide guidance to an industry partner, particularly when they're studying a disease that maybe some companies don't have a lot of expertise in. It also takes input from the regulatory bodies, who have to agree that it is appropriate for a novel agent to move into children relatively early. So in this case, shortly after the completion of the adult phase 1 clinical trial. And then, of course, I can't underestimate the importance of the patient advocacy community to help really push promising agents into the clinic. So with that, I'll acknowledge the patients and families who participate in our clinical trials. It's really so important for us to advance the field. Once we get these agents into the clinic, we need patients and families to participate so that we can learn about these novel targeted therapies. I'll thank my collaborators, industry partners, funding, and then my team back at Dana-Farber. Thank you.
Thanks, Lucas, Jan, and Steve, for a really, really interesting talk. We have some questions. We've got 10 minutes or so, or maybe a little longer, as we've got a break after this session. So I'll start off. We have a question, maybe one for you to kick off, Jan. Do structural abnormalities, 1p deletion, 11q, 17q, offer some kind of targets? Are they actionable events in themselves?
Yeah, it's a good question, of course, because these are the most frequent genomic aberrations in neuroblastoma. And the general idea there is that there's, on those large chromosomal regions, there's multiple genes that for the regions that are gained, like 7q. So there's an extra copy of 7q. There's many genes that the tumor likes a little bit. Yeah, and in the lost regions, there's many genes that tumors dislike. So apparently it is not one single pathway that is activated. It's multiple things at the same time. And that makes it very difficult to target. And we're trying those type of experiments, for instance, with a new CRISPR-Cas9 technique, where we're using cells where we just cut out the 11q region and then compare all the compounds that we have in the cells with 11q deletion and the cells without the 11q deletion. And when we look at the differences, it's not that big. So apparently it's really something subtle that gives an advantage, but it's not really driving one pathway. So there's also not one compound working. So that's indeed a very difficult one. And I think we try to find other ways to find the crucial nodes in the deregulation that you can target. But that's indeed more complex than compared to other tumors. And that also might be a reason why neuroblastoma are so hard to treat.
Thank you very much. I have a question here. This one's from the owner. Incredible way to present this topic. Thanks for your dedication to curing more children and curing them better. What is your biggest frustration, each of you, when it comes to the drug development process? You've got 60 seconds each. Lucas?
Yeah, I mean, I think is the speed. Is everyone's fault or no one's fault, but is the speed that we take, the time that we take to take off the trials, the time that things take. That we have got good ideas, but it is it does take years to make them into a trial. That's the first one.
Steve?
I think for me, it's the it's being told no, I have to say. So we're academic investigators. We are not in the business of making drugs. And so to do these clinical trials, we need a committed industry partner. And as I alluded to with the BET inhibitor story, we're told no a lot. And I think some of that is because we're looking after children, and sometimes that is enough of a disincentive. We're looking after a very rare disease that can be a disincentive, and we're often told no because companies want to see if the drug that they are developing is going to succeed in much more common adult cancers before they're willing to move into a very rare disease. So I think that's my my biggest frustration, particularly when we go to a company with a really strong data package and say, this just makes absolutely perfect sense. And to be told no is very frustrating. There's a glimmer of hope. So at least in the US, we have now the Raise for Children Act, which I think will mean we're told no less.
and there are also things moving forward in Europe and similar. Jan?
Yeah, for me, I think it's the failures. I think there's a lot of trials being done in neuroblastoma that builds a bit up to what Stephen said. There's a lot of trials being done in the past in neuroblastoma which did fail, and that's because a lot of the compounds that were tested were based on adult cancer and developed in adult cancer and then trialed in neuroblastoma without the real proper good preclinical evidence that this is going to work. And I think that is really important, that we make sure that we as scientists and as researchers determine very clearly which compounds work, and then we force companies and pharma to test those combinations of compounds or single compounds in the neuroblastoma patients, of which we have a very good preclinical guess that these can indeed work. Yeah, so that's my frustration.
Thank you. All right, I've got another question here, or a couple of questions I'll try and put together. Targeted drug efficacy question. Neuroblastoma is a heterogeneous disease. Therefore, if a tumor expresses, for example, a target in 60% of the sequenced examined cells, is it considered a suitable target? What about the rest of the cells? Is it unlikely to be affected overall? And then tagged onto that, heterogeneity within metastatic sites, different metastatic sites within the body, or between bone marrow and soft tissue. Does somebody want to comment on those two?
I may just comment very broadly and advocate for combination trials. So we understand absolutely that each patient's neuroblastoma is different, and there's heterogeneity between patients, but within an individual patient, there's heterogeneity, and it's not enough to treat one part of a patient's disease. We need to treat all of their disease, and it's a lot to ask of a single compound to be able to do that. So I think I get very excited about drugs that work in a couple of ways, that are kind of dual inhibitors, or about trials that are bringing together either chemo plus a novel agent, or what I really think we need to do more and more of are trials looking at a novel compound together with a novel compound. We have far too few of those in our field.
Yeah, nothing to add from my side. That's really exactly what is needed. Yeah, completely agree.
Okay, maybe we can move forward with a couple more questions. What if there is resistant tumor after treatment? So I guess we're talking about a tumor that's been biopsy treated and maybe has stable disease. Will it be possible to take a biopsy after the treatment, but before disease progression, to start the precision medicine treatment?
Yeah, that's a good
relapse?
Point. That's a good point. Of course, in some instances there's not complete remission. There's still remaining tumor. From our experience, because we've been biopsying, or we've been taking that material, because it's actually the same material that is taken at operation. And the problem there is that there's a lot of invaded normal stroma, a lot of necrosis. So there might be tumor cells left, but there's a lot of other tissue there. So if you have those biopsies, it's really difficult to do proper genomic profiling of those tumor cells. And so that's really a problem. One thing that we're developing now, which is a technique, this is called single cell sequencing, which is really a brand new technique where you take those pieces of tumor and then really separate all cells separately and sequence them separately. And thereby you can analyze specifically those tumor cells, those few tumor cells that are remaining, and that might indeed give the relapse. So that might be a technique that is promising in that perspective, but that is still all in development and really brand new science. Yeah.
Maybe we've got time for one more. Sorry, Lucas.
I was going to say that of course we do not advocate that all targeted drugs have to be given at relapse. If we find very good drugs, we will want to give them upfront, and that's the example of the crizotinib trial from ZOG, where patients with ALK aberrations from the beginning already get the ALK inhibitor at the beginning, during treatment and as maintenance treatment before they develop a relapse. We don't have yet many very successful targeted drugs in neuroblastoma, but as we have more, surely we will want to bring them up front.
Thank you very much. So I'm going to close the session now, and thanks for the other questions. We will, as we will with all the questions that we failed to get to in the sessions themselves, endeavour to try and feed answers back to you through the website or other means. So there's now a 30-minute break, and we will then be back to you with a parallel session, pain and pain management, and a cell therapy panel session on neuroblastoma. Head back over to the website to join them at the appropriate time. Thanks to our panelists. That was a really great session, and thanks for joining.
Bye-bye. Thank you.
All right, thank you.